So good morning to all of you. So this is our, I think, first lecture on science and technology. Okay, so before we begin the discussion of this portion of general studies, so myself introducing as Dr. Rahul Shankar, and from today's class we are picking up the thread for general science and technology portion. Now, this component of general studies, that is science and technology, including the general science. So compared to the other portions of general studies, this is quite dynamic and highly contemporary in nature. So apart from the concept, you also need to remain highly updated in the context of the exam. So updation means here what is the duration based on which question paper changes every year. So every year how a new question paper appears. So that is particularly, one is the static area, another one is the dynamic area based on current affairs. We'll have the discussion of both together. One is the concept of the topic and followed by the what has happened till date. Beyond that, you have to follow your own the newspaper reading for that particular topic. Now, science and technology, it comes under GS paper 3, under the paper 1, 2, 3 and the 4. It comes on GS paper 3. Some of the questions can be asked in GS paper 2 also, sometimes in 1 also. There will be some overlapping. But overall, it is a part of the GS paper 3. There are certain portions of general studies where questions are asked only for mains exam. There are certain portions of general studies where you get question in both prelims and the mains. So science and technology is one such portion, or one of the portions from where questions will be asked both in prelims and mains exam. And there will be overlapping also again in prelims and mains. So that is why the subject that comes in prelims, so that is the first barrier that you have to clear, the screening test. This is followed by the mains exam. Now, first we look into the roadmap of science and technology, that how we have to approach this subject. This is not any individual discipline, like you study geography or the history. These are individual disciplines, but these science and technology is highly multidisciplinary in nature. It converges the concept of physics also, chemistry also, biology also, applied sciences, which is utilized in the society. So in order to increase awareness, to connect with the society, so now there is rapid technological progresses that we are seeing nowadays. So what developments that have happened for the past 5 years, we cannot have imagined also for the last 500 years. So every year there are some new innovations which are coming, particularly the Nobel Prizes. They give the full overview that what is the area that scientists are focusing. Then, after all, technology is blind in itself. It does not know how to use it, how to apply it. So oversight of human being is required here. So technology cannot take any ethical judgment like human being. Technology, it cannot have empathy or compassion. So that is why technology will require the guidance, the oversight of human being. So that is how we have to connect the technology. So progress of society, it is parallelly along with the technology. Now, first of all, let's see or focus on the syllabus, that what we need to read actually. So prelims followed by the mains exam. If you go through the syllabus in the general studies, the prelims mention the syllabus as general science and mains as science and technology. Now, general science, science and technology both are inseparable from each other. So we need the foundation of general science to discuss on science and technology. Although the syllabus says general science in prelims, science and technology in mains, but remember 90% of questions they will come from science and technology even in prelims. General science, this includes the concepts of physics, chemistry and the biology. Particularly the biology portion will dominate compared to the physics and the chemistry. NCERT textbooks 8th, 9th, 10th, 11th and the 12th. So on any topic, the level of understanding will be minimum of 12th standard. The concept still 12th standard. So these are the NCERT. For 8th, 9th, 10th, we have the individual textbook. For 8th, science book in NCERT. For 9th, individual textbook. These three are combined in one textbook. Whereas in case of 11th and 12th, we have the bifurcation of physics, chemistry and biology portion. Now, regarding the science and technology, before going into the syllabus aspect, what are the references? So one reference is definitely the NCERT textbook. So start now reading from the 8th standard NCERT immediately, without any delay. Just like in history you have to read, isn't it? In history, in fact, you have to read from 6th standard. So start from clean slate, look into flip the pages and start now reading and revising. Minimum 4 to 5 times revision. So you have to look into that, whether you are comfortable or not on the chapters given in 8th standard, 9th, 10th standard. So based on that, you have to decide, you have to underline those sentences that you need to remember. That will help you not only in classroom discussion, following the classroom discussion, but also in the exam also. Now regarding science and technology and definitely science also, what are the other references? Another reference you can write India Yearbook. India Yearbook. For example, chapters on health, environment, agriculture, defense. So here the focus will be on government programs and policies. You will see here that there are listing the government programs and policy related to these areas. So what are the flagship programs of government in the field of agriculture, in the field of defense, in the field of environment, in the field of industry? Third important reference, right? Economic survey. So the latest economic survey. So all these areas are also covered in economic survey. They'll give you up-to-date information, the status, the health status, or the environment, climate change from economic survey. Fourth important reference, the important websites such as website of WHO, website of United Nations Environment Program, website of WHO, World Health Organization, website of United Nations Environment Program, website of International Atomic Energy Agency, website of International Atomic Energy Agency, International Atomic Energy Agency. Then website of India's Ministry of Health and Family Welfare, website of India's Ministry of Health and Family Welfare, website of Ministry of External Affairs, website of Ministry of Environment, Forests and Climate Change, website of Ministry of Environment, Forest and Climate Change. So Contextual reference should be made from these websites also. Suppose you are reading on climate change, then you have to look at this website at what are the up-to-date information provided by the government. Any that is division comes in terms of opinion, that's regarding data. So data you can refer also from the same website. Any discrepancy come, you have to get it corrected from the websites. Then the next one, write Nobel Prizes previous 5 years Nobel Prizes in the field of physics, chemistry and medicine. Physics, chemistry and medicine. Then the yellow book from the Institute on Science and Technology. Yellow book. Then previous 7 years prelims and mains questions for civil services examination. Now this is a very important reference, the point number 7 here, previous 7 years prelims and mains question paper of civil services examination. Because this will help you understand that in what context and how the questions are framed, to what depth examiner can go. So what is the length and breadth of the topic? Some topic that you are reading, but to what extent examiner can go, that will come to know only from these question paper. Your strategy will be based on these question papers. Sometimes question asked in mains will be reflected in the prelims exam. From the same topic, how examiner is able to frame multiple questions, how the statements are framed. So you will get to know only through these question papers. So these are very important reference for your entire journey of preparation. Now, above all these references, whether you are preparing for prelims, preparing for mains, including the personality test or interview, all these three will be under the domain of current affairs. So you can see the importance of current affairs, and current affairs means newspaper reading. So there is no replacement for this newspaper reading by any magazine or any study material, isn't it? Because what today will read in newspaper, how it can be covered in any magazine, isn't it? That information can be asked, and any magazine cannot cover all the complete information of a newspaper. So remember, in entire journey of preparation, you qualify the exam, prelims, mains, interview, improving the rank, all are based on the newspaper reading. Duration in the context of exam, 10 to 11 months before the prelims exam, 10 to 11 months before the mains exam, and interview is highly updated. Same day you will get the question. So that is the importance of newspaper reading, because ultimately whatever you read, it has to be connected to the society, linked to the society, and the most important reference material are the newspaper reading. They keep you updated, they keep you connected with the society. So that is why for an entire journey of preparation, this is the vital source, vital resource. So this will energize you, this will keep you now, will give you buoyancy in this journey of preparation. So you have to be motivated in the context of newspaper reading, that how news are changing, because once you keep on reading, you will also build up your database, which can be converted into knowledge. So you have to process this information that you read from newspaper into knowledge, and then it should be converted into conclusive action. So how you convert this data into knowledge and then into conclusive action to clear the exam. So all depends on your consistency. So consistency will be required. Even if you have missed yesterday's newspaper, you are supposed to read today to finish it off. And while reading, simultaneously try to make notes also. One cannot remember everything. No one is superhuman here. Everybody has to pass through the same route. There are no shortcuts. So same route everyone has to pass through. Number of kilometer journey of the marathon is same for everyone. But the thing is that whether you are starting early, isn't it, in the marathon, and also strategizing your journey. So for this, the strategy will be the newspaper reading. So this should not be missed any day. You can increase the newspaper reading. For example, already communicated to you the Hindu and the Indian Express. So these are the two newspapers that you have to follow. Third, you are able to follow, well and good, otherwise these two are needed. So some news will be repeated, isn't it? So some important events that occur, you should read from both the events. So currently you read in the newspaper about the current events, that how the court is deciding on community resource private property. How do you strike a balance, isn't it? Can individuals' private property is also a community resource? Article 39B. So that debate is going on to be decided by court also. So once you keep on reading this study material, a lot of study material will come. So you are supposed to make notes from the newspaper. Because what you are reading today, you may develop interest in that, but unless until that information, knowledge is there at the time of interview, then only it will be useful. So that is why you are supposed to make notes from the newspaper. If it is handy notes, it can be easily revised. Within 1 to 2 hours you can revise. Even 200 pages notes you may have made in your notebook. It can be revised easily. So revision is also important, but unless you write something, then only we will revise, isn't it? So that is on the aspect of newspaper reading. So these are the important references for our science and technology portion. Now let's see what are the chapters mentioned for the science and technology portion. In prelims they have only mentioned what? General science. So science and technology, first important chapter, biotechnology and issues related to intellectual property rights. Second one, public health. Now public health, this is about the discussion on immunity, diseases, and the vaccines. Regarding diseases, HIV, AIDS, polio, malaria, tuberculosis, Nipah virus, Ebola virus, dengue virus, influenza virus, which includes both bird flu and swine flu. Then we cannot forget the SARS-CoV-2, isn't it? So we all have experience in the real world COVID-19 pandemic, that what exactly is pandemic, how it influences the society. We experience the real world, isn't it? In terms of this SARS-CoV-2 severe acute respiratory syndrome coronavirus 2. So this touched everyone's life, whether India or any other part of the world. So that itself became a topic for the examiner to ask many questions, because the only topic that was discussed at the time of pandemic 2020, 2021, 2022. So these 3 years were overwhelmed by this pandemic because it was affecting our life, not only India and any part of the world. So that is why the entire focus was concentrated on what? Controlling the pandemic. And how we have been able to control the pandemic. What is the factor that enabled us to overcome this pandemic? Yes, the most important tool that enabled us to control it was the vaccine. So here we will focus on the concept of vaccine-induced immunity. Vaccine-induced immunity. That what is a vaccine? How does a vaccine work? Types of vaccine. Now this COVID-19 pandemic, it has introduced altogether a new type of vaccine for the first time in the world. What is that? So what is that, the new type of vaccine that was introduced? Yes, it is for the first time in the world we have seen the use of mRNA vaccine. So this is regarded as a great breakthrough of the entire century, introduction of mRNA vaccine. So developed economies, US, UK, European Union, the major type of vaccine they used to control the pandemic was mRNA vaccine. For the first time it was applied over the society. So that is why whenever something new happens, impacts or has applied, so that becomes a question in the exam. So you may see question 2024 prelims exam, 2024 mains exam, 2025 prelims exam from mRNA vaccine. Directly or indirectly question will definitely come. So those areas which are highly predictable, that question will appear. This means to any depth and breadth examiner can go to ask the question. In initial level it will be easier, but gradually they will pick up, go to a higher level in that area. Nobel Prize Medicine 2023 was awarded on mRNA vaccine. Nobel Prize Medicine 2023 has already been awarded in which area? mRNA vaccine. So most important event that touched our life was COVID-19 pandemic. So details on the SARS-CoV-2 virus, coronavirus, along with the details of the vaccine and its concept, including mRNA vaccine. So that became a very important area of discussion and also for the examiner. Now, apart from this, in case of the public health only, you can write some other diseases like hepatitis also. Hepatitis A, B, C, D and the E. So we have just mentioned what are the most important topics from exam point of view, from where we have seen the questions have been asked. But anytime there could be surprises also. A new question can emerge depending on current affairs. Now I write the third chapter as space and astrophysics. Space and astrophysics. In astrophysics and space, particularly in astrophysics, here we deal with the cosmology or the study of universe. So you can see here that it also includes what? Study of universe. So this means as everything comes under universe only, any question could be asked in the exam, because then you cannot claim this was not there in the syllabus, isn't it? Irrespective of subject matter, any question from any topic can be asked in the exam. You cannot claim, you will not be able to claim this was not there in the syllabus, because the syllabus says you have to read what? Cosmology, study of universe. So that is why you cannot, that is, condense, or you cannot confine to certain chapters, certain contents for this exam. The next chapter I write nanoscience and nanotechnology. It deals with significance of nanoscale. Nanoscale means 1 nanometer to 100 nanometer, and 1 nanometer means 10 to the power minus 9 meter. Next chapter, robotics and artificial intelligence. Types of robot such as industrial robot, humanoid robot, role of drones, which come under unmanned aerial vehicle, UAV, bot, which means virtual robot. So sometimes robot can be simply as some computer program, software program performing a task. So that becomes a virtual robot. If this virtual robot or bot is able to enter into conversation with communication skills like human, to speak in human language, then we call it as the chatbot. So as a chatbot, all of you are aware of the term Alexa, aware of the Chat GPT, Gemini, So sometimes question can come on Gemini. We say that we are currently in the era of artificial intelligence. So this is the buzzword, this is the term that is frequently being used by everyone because it is now influencing each one's life directly or indirectly. We are under the control of now artificial intelligence. We can no longer ignore the influence of AI on our lives. So first of all, what is the USP of AI? That its influence is ever expanding. So what is the core role of artificial intelligence? Decision making. And this is the most important role. So in any task, decision making plays a vital role. So AI can make decisions like human. They can make decisions like human. So this is about emulation of human intelligence. We will discuss in more detail as the branch of AI, machine learning, further branch of machine learning, deep learning. Now decision making cannot be taken without any data. So data is the foundation. But initially it is raw data. How that raw data, as we discussed today, can be converted into knowledge and then into action. So AI, machine learning, deep learning, these are the three important aspects. So currently now we are moving towards what? The application of deep learning. So just like human beings are able to make decisions, similarly, they can also make the decisions. Just like we are able to convert one language into another language, isn't it? So people who are polyglot, who can speak multiple languages. So their brain is able to convert what? One language into another language. Every language had certain words, sentences, the grammar, the syntax, it follows. So can AI also convert one language into another language? So that there is no barrier in communication. So for this, it has to rise to the same level of human intelligence. So there we will discuss the application part in the society of AI. Now within robotics, apart from types of robot, you can also mention components of robot, robotic sensors. So within robotics, components of robots, robotic sensors, and then the application of robots. So this is one such area that you have to remain highly updated in your information, particularly in the field of artificial intelligence. So because unless until we know that what is the latest technology introduced, the latest methods used or processes, then only it can be analyzed for deeper analysis. Now the next chapter, that is the sixth one. Information technology. Information technology. So we also say that we are in the era of digital economy. era of digital economy. Now, to remain in digital economy, we have to remain connected, isn't it? Connected here means the involvement of the internet. So revolution of internet, including evolution of internet, web 1.0, web 2.0, web 3.0. Now some important topics related to this chapter: cloud computing, Internet of Things, IoT, then here also role of AI plus IoT. So what is this combination called? IoT is about any device connected to internet or network, isn't it? Whether your smartphone connected to internet or your smartwatch connected to what? The internet, or your washing machine getting connected with internet. So that is Internet of Things. But the question is, what do you mean by AI plus IoT? What do we call it, this combination? What is that combination? This combination is known as AIoT. Known as what? AIoT, which stands for Artificial Intelligence of Things. And AIoT enabled device that is called as smart device. Called as what? Smart device. In what context we are using the term smart here? The smart here means IoT here will only help gather data from its surroundings based upon the sensor it uses. But that data itself cannot take any decision. To make decision, you have to add brain to this device. Brain added to the device is what? AI. Then it becomes smart device, intelligent device also. It will be no longer only interconnected, interactive device, but also become intelligent to make decisions like human being and become autonomous. Become what? Autonomous. So if you want to develop smart car or autonomous car, autonomous vehicles, examples of what? Smart device with that will use AIoT, smart watch, so they not only track your health, they can even alert you on the risk of like heart attack. They can inform you that you are at risk of heart attack and rush to the hospital. That is the ability of the smartwatches. Then Alexa, all of you know, is also an example of smart device. Alexa is also using AI. Now again, in AI you have seen branches, advanced AI like machine learning. It may not use machine learning, a simple algorithm. So if you want to build smart city, in what context we use the term smart city? So without AI and IoT, you cannot build any smart city. So this will involve the role of smart grids. It will involve the role of intelligent transportation and traffic management. Intelligent transportation and traffic management. Smart cities should increase the safety, reduce the accidents. Smart city should promote sustainability. Which is linked with again reducing your carbon footprint. Reducing the carbon footprint. So our critical infrastructure, including bridges or energy plants, they should be safer, isn't it? They should be able to sense in advance risk of wear and tear, accidents, to avoid that in future. So health monitoring, not the individual health, like smartwatch, even monitoring the health system of like oil and gas industry will be also part of it using the AIoT. Monitoring floods, preventing floods in the cities. So you have to monitor what? The flow of the river, the speed of the river. You have to monitor the level of the river, and you have to generate early warning system about incoming flood. River flood monitoring system, that should be also part of smart city. So whatever you are reading, you have to connect it to the society. You have to develop that concept, the clarity, and connect yourself with the society, how the technology will be applied. So that is why this is a very important chapter, information technology. This also includes discussion on blockchain technology. Blockchain technology is also part of it. Now the essence of blockchain technology is about that how one cannot tamper with the data. If data cannot be tampered, then that platform is providing me blockchain technology. So data once saved cannot be changed in blockchain technology. It provides me data fidelity. It provides me immutability. Then come the other characteristics like decentralization, distributed ledger system followed in the blockchain technology. So how do we connect blockchain technology with our governance, to maintain the sanctity of data, avoiding tampering in land records, avoiding tampering in voting also. So why we are not able to go for electronic voting currently? That it is individual may not come to the physical booth and can vote from anywhere to electronic voting. So that can be done only when it is done on the platform of blockchain technology. So Election Commission of India has already collaborated with the IIT Madras on the blockchain technology introduction. So therefore, this is again another important chapter that we must remain highly updated in this and its application part. Then write the 7th chapter. 7th chapter is about indigenization of technology. Indigenization of technology. Indigenization means here homemade, something which is made on your own. Homemade products and services, homemade technology. That is indigenization. So here the focus is on Make in India, in defense, Make in India in health and medicine to make the vaccines, in the field of agriculture, manufacturing of semiconductors, indigenization in making satellites, launch vehicles, and the outer space missions. It does not mean we are closing the doors against globalization. The doors and windows are open. It will follow global collaboration, collaborative approach. So we have to learn from others, we have to collaborate with others. So indigenization of technology along with the global collaboration, where scientists, policy makers of the world, so that there could be innovation. So focus should be on what? Innovation. But this innovation should be with equity. Innovation with what? Equity. That is the most important aspect. So you are introducing some new technology, but whether it is able to bridge the gap, whether it is able to promote prosperity in the society. If there is lack of prosperity in the society, then that innovation is of no use to the society. So innovation should try to increase per capita income in the country. It should try to increase the quality of life of individuals, irrespective of race, religion, caste, sex and border. Innovation itself will not decide. Policy makers, they have to come in forefront that how that policy is going to benefit everyone. Then the next chapter, contribution of Indian scientists. So the eminent scientists of India and their focus areas, how they have institutionalized science and technology in India. So for example, Homi Bhabha, so he is said to be the father of India's nuclear program, father of India's nuclear program. All of you know about the Dr. APJ Abul Kalam. Then similarly, Vikram Sarabhai. So eminent scientists of India, so what has been their contribution in the specific areas? So whether it is nuclear program, the space technology, missile program, so any individual scientist could be asked or their focus areas, that how they have made a huge impact for either for security of the country, or they have institutionalized innovation with equity. So you have to focus on that their contribution resulted in innovation with what? Equity. So you have to connect science and technology as part of innovation, but how you are going to promote equity in the society. That is the most important aspect. So these are the few, but the important chapters, the roadmap to science and technology. What should be your action plan from today's class in the course of science and technology? In today's class, we are going to begin the discussion with the public health. So we are going to start now with the public health. So first we are going to introduce certain words, keywords which will be used in the discussion in the entire class and further, and then we'll start the exact discussion on the topic. So let's introduce. Do we need to read all the three domains of physics, chemistry, biology, class 11, 12? So there is no defined syllabus here. Whatever we are discussing in the class covered in NCERT, you have to read that. So you have to go along with the class and also based on the previous year question paper. So in public health, write down the term first, immunity. Immunity means ability of the body to fight against infections and diseases. Ability of the body to fight against infections and diseases. Or you can write simply as disease resistance. Write as disease resistance. Now write the term pathogen. Pathogen means disease causing microbe or microorganism, disease causing microbe or microorganism, for example bacteria, virus, then write the next term, antimicrobial medicine. Antimicrobial medicine. Is the medicine used to prevent the growth and multiplication? Prevent the growth and multiplication or to kill the pathogen? Or to kill the pathogen? For example, antibiotics are used to prevent the growth and multiplication, or to kill the bacteria, Antibiotics are antibacterial medicine. Antibiotics are antibacterial medicine. Antibacterial medicine. Now, suppose all of you, for example, knows paracetamol, isn't it? Is paracetamol antimicrobial medicine? Is paracetamol antimicrobial medicine? See, antimicrobial medicine will also include antiviral drugs. This is also under what? Antimicrobial medicine. Antiviral drugs, they are going to prevent the growth and multiplication of the virus. At the same time, virus is resistant to antibiotics. Virus is resistant to antibiotics. Is it correct statement? Is virus resistant to antibiotics? What is your answer? Virus is resistant to antibiotics. Is this a correct statement? So these are statements may come in the prelims exam from this basic science, general science. This is the correct statement. Antibiotics, they cannot prevent the growth and multiplication of virus. They can only act against what? Bacteria. But you have seen that in viral infection, doctor may prescribe you antibiotics. What is the reason? The reason is to control, so virus infection may suppress your immunity, and that suppressed immunity may be vulnerable to secondary bacterial infections. So to avoid the complications of secondary bacterial infection, doctor prescribes you antibiotics. But there is no direct role of antibiotic on the virus. In fact, there is no role of antibiotic against the virus. Virus is completely resistant to antibiotics. Your own immunity has to fight against the virus mostly. So mostly if you see like in dengue virus infection, any viral infections, you are mainly dependent on what? The paracetamol to control the body temperature. Paracetamol you are taking as antipyretics and analgesic. So paracetamol is what? Antipyretic and analgesic. Antipyretic means which will lower the body temperature. Lower the body temperature. And analgesic means pain reliever. It will reduce the pain reliever. Paracetamol cannot prevent the growth and multiplication of bacteria virus. Is it clear? So paracetamol is not antimicrobial medicine. Antibacterial medicine means which will act against, whether it is acting against bacteria, virus, fungus, protozoa, some disease causing microbe. Paracetamol only will act on the symptoms, because these symptoms could be there during infection or diseases. Now recently there was a news on Singapore and Hong Kong banning the spices from India of Everest and the MDH. What was the reason? Presence of, yes, ethylene oxide. So ethylene oxide used in Indian spices. So what is the reason that India has to use ethylene oxide? Yeah, that is basically a preservative. It preserves. So it will destroy the microbes. So any bacteria, any fungus that may be present. So they used as what? Ethylene oxide is used as what? Pesticide. Ethylene oxide is present as pesticide. And because being used as pesticide, ethylene oxide is said to be carcinogenic or cancer causing impact. So long term exposure, prolonged exposure, as it destroys microbes, it will preserve your spices for a longer duration. So this is used to mainly what? To enhance the shelf life. So during preparation, in order to enhance the shelf life of the product, we are using it. But unfortunately, it is nothing but a pesticide, and long-term exposure or prolonged exposure, so that makes it what? Carcinogenic. Cancer causing impact is there. So that is why it was in news, ethylene oxide. So sometimes questions could be asked, as you can see, based on current affairs related to the science. Now coming back to this immunity once again, let's focus on the term virus in more detail. Now, what are the building blocks to make up a virus? What virus is made up of? Genetic material plus protein coat called capsid. So the two important building blocks for a virus includes protein coat and the genetic material. These are two important building blocks of a virus. Now, any virus for its multiplication, reproduction, it needs the shelter of a host cell. Suppose this is the virus, and now we are showing or depicting the virus multiplication. Virus replication. Replication means multiplication. So we are showing here that a virus is multiplying inside a host cell. Now what do you mean by host cell? Host cell is giving shelter. It is giving what? Shelter to this virus. Virus is performing its biological activities inside the host cell. Virus is getting the benefit, isn't it, from this host cell? What is the benefit the host is getting from the virus? Can you tell me? This host cell can be a plant cell. Host cell can be a plant cell. It can be an animal cell. It can be even a bacterial cell or bacteria. Fungus or fungal cell. Now the question is, virus is getting the benefit from the host, isn't it? It is using the machinery of host cell for multiplication to perform its life activities. But what is the benefit host cell is getting from the virus? So this relationship shows that host is not getting any benefit here. Virus is only getting the benefit. So relationship between two living species where one gets the benefit, isn't it? Another does not get any benefit. So this virus is acting as a parasite. Acting as what? Parasite on this host. So virus is acting as a parasite. Now the same virus outside the host cell, can it multiply? Outside the host cell it remains dormant. So outside the host cell virus is what? Dormant. Virus cannot perform any of its life activities outside the host cell. Can protozoa be a host cell? See, animal cell includes amoeba also. Is it clear? Amoeba is what? A protozoa. Unicellular organism. That is the protozoa. Multicellular also can act as a host for the virus. So animal cell includes here amoeba also, human body cell also, or any other animal body. Birds also. So virus outside the host cell, that is host cell, remains what? Dormant. It cannot multiply. It will multiply only inside the host cell. This applies to any virus. So is it that we can say that all viruses are parasite? Is it correct? All viruses are parasite. Virus is defined as an obligate intracellular parasite. Virus is defined as what? Obligate intracellular parasite. Parasite because it is deriving benefit from the host, not giving any benefit to the host. Intracellular means inside the host cell. Intracellular means what? Inside the host cell here. Obligate means compulsory. So for any virus, it is compulsory to take shelter into the host cell for its multiplication, for its life activities. So combining all these characteristics together, we define virus as an obligate intracellular parasite. Or we can also say that virus is an intracellular microbe. Intracellular microbe. So obligate intracellular parasite, or we can simply say virus is an intracellular microbe. So microbe means what? Microorganism, the one that you cannot see with your naked eyes. You need some form of microscope. Obligate means compulsory. It is mandatory for any virus to take shelter into host cell to perform life processes. Now virus host cell interaction. There is another point here, and that is virus host cell interaction. It follows the property called host specificity. Host specificity. Host specificity means for every virus, virus has evolved to select a specific host where it will attach and enter and multiply in the host cell. For every virus, there is a specific host. Although we have written here, virus can use plant cell also as a host cell, animal cell also. It is not that the same virus is using all the 4 different types. Every virus has its own specific host to which it will attach, enter, and will multiply inside the host cell. So this means virus is able to identify its specific host cell, where it has to land, to attach, enter and multiply. We will take the example of coronavirus, SARS-CoV-2 virus, to discuss this. Now, coronavirus, we have experienced that this is, or this causes respiratory infection. And route of transmission is through inhalation of respiratory droplets So when infected person coughing and sneezing, producing respiratory droplets that may contain the virus, and this were inhaling. So this enters via the nasal cavity, isn't it? It enters via your nasal cavity, and then it can reach your lungs also through inhalation. So this enters via the nasal cavity. Therefore, we say that it is causing respiratory infections, respiratory tract infections. Today we discussed that virus building block includes what? Genetic material and the protein coat or capsid. Now, virus based on genetic material it can be a DNA virus, deoxyribonucleic acid, or it can be RNA virus. So genetic material is either DNA or RNA. What is mean by genetic material? Genetic material means that acts as software to produce the new elements of the living organism. So for example, this DNA is the complete software of the DNA virus. How that virus will multiply, what are the viral proteins that needs to be produced, all that information, biological data will be present in the DNA. So DNA contains what? The complete biological information, biological data acting as software to produce the complete organism. So this virus DNA will have the complete information data which will be used for production of new viruses in the next generation. Similarly, in case of RNA virus also, here RNA is acting as the biological data information source to produce new viruses, new copies in the next generation. A DNA virus, for example, so what are the important examples of DNA virus? Suppose this is the genetic material DNA and this is the protein coat that we discussed, or the capsid. So capsid is giving protection to the genetic material of the virus. Now what are the examples of DNA virus? So examples also should be taken in the context of exam or the relevance to the society, applied to the society or affecting the society, impact on the society. So under that we can write some examples like hepatitis B virus, human papilloma virus, so one example is hepatitis B virus, second example is human papilloma virus, where we have written it causes genital cancer. For example, cervix cancer in females. But we have the vaccine to prevent it. So females around 10 to 11 years are vaccinated using HPV vaccine to prevent this. So this is an example of virus-induced cancer. Just now you have written what? Ethylene oxide as a pesticide can cause what? Cancer. Cancer can also happen because of what? Virus also. So there are several reasons for cancer. One of the reasons virus-induced cancer, another could be pesticide-induced cancer. Now, how statements come in exam? What is the relevance? Suppose they ask you that human papilloma virus, it can cause cancer in both males and females. Is it a correct statement? This is how statements are asked in the exam. Yes, it can cause penile cancer or genital cancer in males also. Human papilloma virus. Can hepatitis B virus cause cancer? What is your answer? Can hepatitis B cause cancer? Yes? Liver cancer. So there is a risk of liver cancer because of cirrhosis, followed by liver cancer because of hepatitis B also. So this also increases the risk of which type of cancer? Liver cancer. So that is why these are not mentioned in the syllabus. When you compare history, geography, polity with the science and technology, so area is undefined, highly scattered information. So first we have to define the name of the topic and then we have to discuss the topic. In other subject, these are single discipline where it is predefined, name of the topic. Half work is done there. You know what to read there. But here you do not know what to read. First you have to identify what you need to read and then revise, and then you will start acting. So that is the difference in other subjects in science and technology. Another important example is adenovirus. Adenovirus. Now, why we have taken adenovirus as an example? Because it has a special relevance in developing viral vector vaccines. Used to develop viral vector vaccine. For example, all of you know about the Covishield, isn't it? COVID-19 vaccine, Covishield. So that is an example of viral vector vaccine. So to introduce this vaccine into the body, we have to inject what? This adenovirus into the human body. So this was the major vaccine used by India to control the COVID-19 pandemic. Serum Institute of India produced this vaccine for us. So this is an example of COVID-19 vaccine. But under which type of vaccine? Viral vector vaccine. So ingredients of the vaccine carried by this virus. There are different types of vaccine, but just to make you understand what is the relevance of adenovirus. Then another example you can write pox viruses, including monkeypox virus and also the smallpox. Have you heard about LSD virus? LSD virus. lumpy skin disease virus. But this infects only the animals, infects the cattle. This is non-zoonotic. Zoonotic means that there is a risk of transmission of disease from animal to human. From animal, it mainly infects animal, but it can spill over to human, like Nipah virus, Ebola virus, influenza virus, bird flu. But this is what non-zoonotic. Non-zoonotic means it is confined to animals only. It is not infecting the human. Still, indirectly it can affect human life. How it can affect human life? Because this is affecting human livelihood. Because this is a fatal disease. This can reduce milk yield in cows. It can cause infertility in cows. It causes death of cattle. Causes the death of cattle. So because of these reasons, entire cattle industry gets affected due to LSD. You have to stop movement of cattle altogether to avoid the risk of spread of disease. If you stop the movement of cattle, the cattle industry gets affected. So automatically small marginal farmers, dairy farmers who are part of this cattle trade, their livelihood gets affected. And there is fast transmission, cattle to cattle, of this LSD virus. So sometimes questions could be asked based on current affairs. So 1 or 2 years back, there was fast spread of LSD virus in India. In southern states also, northern states also. So these are some important examples of what? DNA virus. Now let's take some examples of RNA virus. First I will write coronavirus, SARS-CoV-2. Coronavirus SARS-CoV-2. Already I have written the expansions, severe acute respiratory syndrome, coronavirus 2. Second example, influenza virus. Third example, Ebola virus. Fourth, Nipah virus. Fifth, dengue virus. So these are some important examples of RNA viruses. Now, which was India's first indigenous COVID-19 vaccine? What is the answer? India's first indigenous homemade COVID-19 vaccine. So first indigenous COVID-19 vaccine of India. Answer is Covaxin. So Bharat Biotech introduced this co-vaccine. This contains inactivated or non-replicating SARS-CoV-2 virus. So ingredient present in this vaccine contains dead or inactivated or non-replicating SARS-CoV-2 virus. So one was Covishield, another one is what? Co-vaccine. Both are COVID-19 vaccine. But co-vaccine is the India's first indigenous homemade COVID-19 vaccine. So you have to remember facts also, concept also on the topic, analysis of the topic, applied part of the topic. So we are writing examples of RNA viruses where RNA is the genetic material present in the virus. In fact, when you compare DNA virus with RNA virus, which is more unstable to undergo faster genetic changes? RNA virus. So RNA viruses, they are more unstable. More unstable means there is, so these are more unstable and increased risk of mutation. Mutation means genetic changes. So genetic changes seen are faster in RNA virus compared to the DNA virus. DNA virus is a relatively stable virus. So risk of a virus jumping from animal to human body. Jumping from what? Jumping and means spillover of the virus. Spillover, we use the term. If a virus is able to spill over from animal to human body, we call it a zoonotic spillover. And that is mainly because of deforestation. Increasing human-animal environment interaction increases the risk. So RNA virus, which is more unstable, there is a higher risk of converting from animal to also human infection. Like Ebola virus or your Nipah virus, these are examples of what? Zoonotic viruses that have spilled over from animal to human body. So it is difficult to control these RNA viruses, difficult to control these RNA virus. So even if vaccines were introduced, we are able to control the pandemic, but are we able to eliminate this coronavirus? Can we eliminate coronavirus? No, because the virus that undergoes fast mutation, you cannot eliminate it, you can control it. You can control it. You can prevent morbidity, mortality in the society. Still infections could be there of this coronavirus, including the SARS-CoV-2. Now, before we discuss more on the virus part and this genetic material, we will discuss later in detail that what are the chemical composition of DNA. So both are genetic material, but universal genetic material, plant, animal, bacteria, we have what? DNA as the genetic material. Only in virus, either DNA or the RNA genetic material. So what we will do now, we are going to discuss first on difference between bacteria and the virus. So that we have more clarity in the advanced discussion. So bacteria described as extracellular microbe, and virus we defined as intracellular microbe. So unlike virus, bacteria does not need a host cell to take shelter, isn't it? Bacteria is capable of independent multiplication or self multiplication. So bacteria can multiply on its own. It does not require any host cell to enter and then multiply, unlike a virus. Virus always needs a living host cell for its replication or reproduction. So today we discuss what? The host cell and the virus. Host cell that is giving shelter to the virus. Virus is using the host cell for its multiplication, reproduction. So virus always needs a living host cell for its replication or multiplication. But there is self-multiplication in bacteria. Now, any living organism for its survival requires energy. And how this energy is produced in our body? Our food is getting converted into energy, isn't it? So there is a biochemical process which is present in our body that converts food into energy. What do we call it? Biochemical process that converts food into energy. Respiration. Answer is what? Respiration. For survival of any living organism, respiration is vital, isn't it? Otherwise, how it is going to derive energy to perform its life activities? So, is respiration found in a virus? What is your answer? What type of respiration? If you look into bacteria, respiration in bacteria can be aerobic also, anaerobic also. So in presence of oxygen, it will undergo aerobic respiration. In absence of oxygen, it can even undergo respiration called anaerobic respiration. But whatever respiration we will write for a virus. So whatever respiration is there in a virus, aerobic or anaerobic? Yes? There is no respiration in a virus. This is the answer. So virus is not capable of respiration. It cannot convert any food into energy. It cannot produce its own energy source. It is totally dependent on its host cell. So what we will write here is virus derives energy from the respiration of its living host cell. So virus derives energy from the respiration of its living host cell. To perform its life processes. To perform its life processes. Now, we all know that bacteria can decompose. It acts as a decomposer, isn't it? It can break down dead decaying bacteria. So, as an important decomposer in ecosystem, bacteria plays a vital role in recycling the nutrients in the soil. Can we use virus as a decomposer? What is your answer? Can virus act as decomposer in ecosystem, contributing to environment? So as a decomposer, remember, one is the fungus, another one is what? Bacteria. Decomposer in ecosystem, not the virus. So virus does not have enzymes. For example, fungus has enzymes which will help in decomposing dead and decaying. But virus does not have any such chemical enzyme in order to decompose. So it cannot act as a decomposer in ecosystem. Coming back to bacteria once again, genetic material always DNA in a bacterial cell. So genetic material is always DNA, and genetic material in a virus, just now we have written, either it is DNA or RNA. The next important basic difference between bacteria and virus we can write is regarding the culture medium. Culture medium or nutrient medium. So if we want to study these virus bacteria in this open air, we cannot study them. We have to bring them into lab, isn't it? We have to culture them into lab. So to culture them in lab means what? To make them grow and multiply in the lab. So what is that culture medium, nutrient medium required for the growth and multiplication of bacteria, for the growth and multiplication of virus, in order to collect important characteristics of this bacteria and virus? How do we go for drug testing? That which type of drug will act against this bacteria or a particular virus? So for all these we have to bring them into lab conditions. We have to study that what is the environmental condition needed by them to multiply, which environmental condition can restrict their growth and multiplication. So for this we need the help of these culture medium. So their biological studies, characteristics collected through what? Using culture medium. Now culture medium or nutrient medium for bacteria, we can use non-living medium. We can use what? Non-living medium. For example, you can write nutrient agar, and agar means here it is the complex carbohydrate or polysaccharide. Agar is chemically a complex carbohydrate. Now bacteria is capable of converting what? Food into energy. Is it clear? So we can use nutrient medium or nutrient agar, which is a non-living medium. It is not that bacteria cannot grow in living medium. Bacteria in millions, billions, trillions find inside your body in a living medium, isn't it? What we are seeing as a culture medium in the lab, we can use non-living medium to culture the bacteria. What about the virus culture medium? Always use living medium. For virus as a culture medium in the lab, we have to use always what? Living medium. So for example, lab animal. This could be a monkey, it could be lab rats, isn't it? Albino mice, guinea pigs. So we are putting the virus inside their body, inoculating the virus inside the body of animal, the living animal. Although there are ethical concerns, if it causes death of this living animal, then there are ethical concerns that should be avoided. Then it can also be embryonated egg. So we can see here hen's egg inside which there is a living embryo, and you are putting the virus into this living embryo. And then we will gather a data that how this virus, what changes it will cause, biological changes, physiological changes. Will it cause some blood clots? It will damage some liver cell, a kidney cell. We can also test drugs, antiviral drugs. We can also go for clinical trial of vaccines. So for vaccine there is preclinical trial using what? Lab animals. And then we go for clinical trial using the human body. So preclinical trial is vital. Directly we cannot select a human for trial of a vaccine. We have to understand first whether it is toxic, extremely harmful by taking human-like physiology or the body. So that is how hen's egg, for example, influenza virus, bird flu virus. First we are testing in what? The antiviral drug or the vaccine in the hen's egg to study the virus. But these are all living medium, isn't it? So for a virus, we cannot use any synthetic or artificial medium. You can write this. Write a note and then note down virus always needs a living medium for its growth and multiplication. As a culture medium. For its growth and multiplication as a culture medium. Next sentence. Any artificial or synthetic medium, or non-living medium any artificial or synthetic medium or non-living medium cannot be used as a culture medium for a virus. Cannot be used as a culture medium for a virus. So these are the differences between the bacteria and the virus. Now still there is another important point about the virus. Virus is said to be having both living property and non-living property. So one leg of virus is in the living world, another in the non-living world. Today we discussed that there is no respiration in a virus. Can you tell me a living organism where there is no respiration? Virus, there is no respiration, isn't it? Can you tell me a living organism where there is no respiration, which cannot convert its food into energy? So there is no any such living organism. And that makes the virus acting as what? Non-living, isn't it? In fact, virus outside the host cell cannot perform any life activities. So that is why it has both living properties and also non-living properties. So virus is regarded as at the transition phase or at the border of living and non-living. You can write this point. Virus is regarded as at the transition phase. Virus is regarded as the transition phase of living and non-living. Transition phase of both living and non-living, as it has both living and non-living properties. Then write, viruses are obligate parasites. Viruses are obligate parasites that can reproduce, obligate parasites that can reproduce only by infecting, that can reproduce only by infecting living host cells. Now we come back to our discussion on virus host cell interaction once again, and taking the same example as SARS-CoV-2 virus. So we have drawn here SARS-CoV-2 virus, where the genetic material is the RNA, and this is the spike protein of the virus. Today we discussed that vaccine, for example, Covaxin, when given to the body develops vaccine-induced immunity. Body develops what? Vaccine-induced immunity. Now, what is this as a part of vaccine-induced immunity that provides us protection from this virus? So vaccine will help develop what? Memory cells in your body. So when you are vaccinated, then your body develops what? Memory cells. These are specialized lymphocytes. These are type of white blood cell only. So these are specific type of white blood cell that transform into what? Memory cell. So they develop the memory of what? The protein of the virus. They develop the memory of what? Protein of the virus. What is that protein present in the virus? Spike protein. When you are injected with the vaccine, vaccine carries what? The virus into your body. It carries the spike protein of the virus. And the body starts learning so this co-vaccine today we discussed, it contains non-replicating SARS-CoV-2 virus. This is the ingredient present in the vaccine. So it contains the protein of the virus, spike protein. Corresponding to the spike protein, the body develops what? The immunological memory of the protein of the virus. And based on that, body can produce specific antibodies against the virus. These Y-shaped structures are what? Antibodies. In the context, we have written anti-SARS-CoV-2 antibodies. So they will be able to target the spike protein of the virus. But before we discuss how they are going to target this virus, first let's discuss when the virus enters. So today we discussed that when you are inhaling the virus through infected respiratory droplets, it enters your respiratory tract and can reach the lung cells. So human lung cell So we have drawn here virus-host cell interaction. This virus-host cell interaction is based on the receptors present over the host cell. So this is the host cell. This is the host cell, or this can act as a host cell for the virus. So when the virus enters your body, then it will try to attach to your lung cell. And at sticking point, the stickiness to make the virus to attach or stick to your lung cell is the presence of ACE2 receptor. So these ACE2 receptors, they act as door for the entry of the virus. So virus spike protein will stick to these receptors, attach to these receptors, and then it will try to gain entry into the lung cell. If it is able to gain entry, means what? It has infected the body now and is multiplying in the lung cell. Multiplies in lung cell, it will cause inflammation of lungs, breathlessness symptom will come now. This is what was happening during the COVID-19 pandemic. However, if a person has been vaccinated, this interaction can be blocked. Interaction can be blocked. Before go to that level, if you remember, during COVID-19 pandemic, more and more adults were getting breathlessness and hospitalized rather than children. What was the reason? Is that children, they have a better immunity, 2 years old infant than 20-year-old adult? Whose immunity is better? 20-year or 2 years? So what was the reason then that 20-year were getting breathlessness, hospitalized rather than 2-year-old child? What was the reason? Initial days, suppose the year 2020. In 2020 there was no vaccine, isn't it? So online they are getting the correct answer, non-developed lung cell. So lung cells of infants are yet to be fully mature or developed in terms of ACE2 receptor. So immature lung cell does not have those numbers of ACE2 receptor that will enable strong attachment to the virus. So virus was loosely sticking to the infant lung cell, children lung cell, and was strongly sticking to the lungs of whom? Adults, because of more number and concentration of developed ACE2 receptor over lung cell. That was the reason. Is it clear? So these ACE2 receptors, they are in numbers, in more concentration present around the adult lung cell rather than the infant lung cell. So therefore virus was strongly sticking to whom? Adult lung cell rather than the children lung cell, and that was the reason for higher intensity of impact, higher viral load in case of adults rather than children. So immature lungs of children was acting as a blessing in disguise to prevent attachment of the virus and avoid at risk of breathlessness and hospitalization. That was the reason. Coming back to here, so once you are vaccinated, then your body produces what? Suppose this is the real virus. After vaccination, the real virus enters your body. So you can see here these antibodies, they block the virus attachment or infection. So what you've written, these antibodies, they are blocking the spike protein. They will not allow the spike protein of virus to interact with the ACE2 receptor of human lung cell. By blocking this interaction, they cannot attach, they cannot enter, and indirectly they are forced to leave the body. It will help in viral clearance. So antibody blocking the virus attachment, blocking the virus infection. So this is an example of antibody mediated immunity. Antibody mediated immunity. The same can be drawn here also, isn't it? So these antibodies They are blocking the interaction of virus with the ACE2 receptor. So this is our antibody mediated protection that enables the individual to provide protection from the virus infection. Although we'll discuss more on the vaccine, that vaccine role is to prevent disease, not necessarily infection, because there are many vaccinated people who became infected but were prevented from severity and hospitalization and deaths. So even after vaccine, there could be a risk of being infected, but it will definitely prevent what? Severity of the disease and hospitalization. So that is on vaccine-induced immunity. Now these ACE2 receptors, are they present only to attract the virus, stick the virus, or they have some biological role in human body? So what is the biological role of ACE2 receptor in human body? This ACE2 receptor, they help in maintaining your blood pressure. Help in maintaining what? Blood pressure in your body. They are present not only over the human lung cell, these are also present over the kidney cells. These are also present over the heart muscle cell, and together they help regulate your blood pressure. So that is the natural role, physiological role of the ACE2 receptor. But unfortunately they act as sticking point for the virus. So this was an example of virus host cell interaction. For example, HIV virus enters the body. Will it use the same door for its entry? What is your answer? What will be the door for HIV then? What is the door for the entry of HIV virus? Just like we have written ACE2 receptor present over lung cell. Again I repeat the question, what is the door for the entry of the virus? This door will be present over some cell. What is that cell? HIV virus will stick to what? A blood cell. Which blood cell? White blood cell. So whenever there is involvement of white blood cell contains the exact door for attachment and entry of the HIV virus. White blood cell. Therefore, involvement of white blood cell due to any reason, that can increase the risk of what? Transmission of HIV infection. So every virus has its own door to attach and enter. It is not necessarily that the same door used by all the viruses. So in virus host cell interaction also there are specific receptors which will enable the virus to enter the host cell. It depends upon the evolution of the virus, the nature of the virus, the type of the virus that it comes across during our lifetime. So this is overall the example of virus host cell interaction and the concept of receptor over the host cell. Now, the term bacteriophage, write down, bacteriophage is a virus that infects the bacteria. Bacteriophage is a virus that infects a bacterial cell or bacteria, that infects a bacterial cell or bacteria. Which means bacterial cell acting as a host cell, which means bacterial cell acting as a host cell for bacteriophage viruses. Bacterial cell acting as host cell for bacteriophage viruses. Now, it is not that all bacteria are disease causing. Some bacteria could be good bacteria also, isn't it? So, we have a concept called gut microbiota. Concept called gut microbiota. Now, gut microbiota, it boosts the intestinal or gut immunity. It will boost our intestinal or gut immunity. Replaces pathogenic or disease causing bacteria. These are beneficial microbes including bacteria, lactobacillus, bifidobacterium. These are some examples of good bacteria. All of you know the name lactobacillus. Another important name is bifidobacterium also. Now these include intestinal bacteria, present in small intestine and large intestine. Now the question is that within this gut microbiota, when you compare small intestine with the large intestine, where we have more in number and diversity, in which part of GIT? So gut means here your gastrointestinal tract. Gut means gastrointestinal tract or GIT. So once again the same question. Per ml, the number of bacteria is higher in small intestine or large intestine? Answer is large intestine, not the small intestine. So per ml, in case of small intestine, it is in millions. Per ml, in case of large intestine, it is in billions and trillions. So greater number diversity, gut microbiota is present in the large intestine, not the small intestine. It is present there in millions, here it is billions and trillions. per ml fluid that you take from the intestine, per ml the fluid that you take from that intestine. Based on that we are saying it is millions, billions and the trillions. And in large intestine, example you can write colon bacteria. Colon bacteria. Colon is a part of large intestine. So we have cecum, colon, rectum in the large intestine. So colon part of large intestine harbors the maximum in terms of the large intestine bacteria, colon bacteria. So they boost our immunity. They will not allow disease causing bacteria to settle in the body. So therefore they modulate our immunity. Now beneficial microbes, including you have written lactobacillus bifidobacterium, these are also called as what? Probiotics. These beneficial bacteria that you have written, these are also part of probiotics. So what are probiotics? The same, beneficial microbes including bacteria, lactobacillus bifidobacterium, it is the same, probiotics. Now there is another point here, and that is prebiotics. Prebiotics, these are the nutrition in the form of indigestible fiber needed for the growth and multiplication of probiotics or beneficial microbes derived from fruits and vegetables. So we need the help of what? Beneficial bacteria to boost our immunity. At the same time, probiotics or beneficial bacteria that you have written, for their growth and multiplication, you have to support them with what? Prebiotics. Prebiotics means indigestible fiber present in the fruits and vegetables. They act as nutrition of the probiotics. So this is the importance of gut microbiota. And this gut microbiota helps maintain gut health, or gut immunity. It's the same. So when we are going to discuss that factors influencing the immunity of the body, there we will once again bring this concept of gut health, gut immunity. But at least you remember these points in the context of gut microbiota. Now write the next heading. Composition of blood. Composition of blood. So we are discussing on immunity. So where we have to discuss on some basic composition of blood, and then we can jump on the role of the immunity. No, fumigation means you are clearing the pests. Fumigation means to clear the pest. You can use anywhere as a pesticide. That is the meaning. The use of pesticide to clear the pest, that becomes fumigation. So use as fumigating agent.
Fumigate was the word.
Yes, fumigant. That's what I told you. To clear the pest, that process is called fumigation. So ethylene oxide will kill the microbes present in the spices to increase the shelf life. So we have written basic composition of blood that you are supposed to remember, so that when we discuss, it is clear to you the contextual discussion. So blood is defined as a fluid connective tissue. Fluid connective tissue. Fluid part of the blood, that is called plasma. Fluid part of the blood is called plasma. And in the fluid plasma are suspended the blood cells. 3 types: erythrocyte, the scientific name of red blood cell; thrombocytes, the scientific name of blood platelets; leukocytes, scientific name of white blood cell. Then the blood plasma, 92% is water only, and there is 8% includes, you can see the composition: plasma protein, nutrients, gases, minerals. Now, as plasma protein, some examples are mentioned here, like globulin. All these plasma proteins mentioned here, whether globulin, fibrinogen, prothrombin, these are produced by our liver cells. So liver is the factory to produce these plasma proteins. Globulin is used to synthesize what? So this is the raw material to make antibodies in your body. Today we discuss antibody-mediated immunity. Our own body is producing it. But to produce these antibodies, our body requires what? Plasma protein called globulin. And that provides immunity to fight against infection, disease. And that is why antibody is also called immunoglobulin. Antibody also called what? Immunoglobulin, represented by capital I, small g. Antibody you can represent as capital A, small b for antibody. So this is the role of this particular plasma protein, globulin. Then the other two written here, prothrombin, fibrinogen. These are blood clotting proteins. So whenever there is some injury to the body due to any reason, and there is a risk of bleeding or blood loss, then the body activates a self-defense mechanism called blood clotting to prevent excessive bleeding or hemorrhage. So to prevent that excessive bleeding, body has to form what? Blood clot. For the formation of blood clot, to block that bleeding, the body requires these blood clotting proteins. The two essential one are prothrombin and fibrinogen. So these are raw material to form what? The final blood clot. There are also other ingredients like calcium is also required for the formation of blood clot. Platelets are also required because at the site of injury, platelets, they release the signal to initiate the process of blood clotting. At the site of injury, platelets are activated to release a chemical signal to initiate the process of blood clotting. So it is a coordinated process, coordinated effort by the body involving platelets, blood clotting protein, calcium to form the final blood clot and prevent that bleeding. Nutrients include glucose, amino acids. So blood is an important transporting medium. It transports. So how does glucose reach the cells, the liver cell, kidney cell, so that it can be broken down and converted into energy through respiration that we discussed. The blood will transport that glucose. How does oxygen will reach there? Because without oxygen, glucose cannot be oxidized. So respiration means oxidation of glucose, isn't it? To produce energy. That biochemical process that we discussed, aerobic respiration. So it is the blood that is transporting the oxygen also. And that oxygen reaches the cells to convert or break down food into energy. So therefore blood is an important transporting medium to transport glucose. Amino acids means these are building block of proteins. Gases including carbon dioxide, oxygen. Minerals, example Na sodium, K potassium and Ca calcium. So minerals are also present here. So this is the basic composition of our blood. So till now we tried to focus on gut microbiota, we focused on immunity. Now we have to continue further by taking the example of blood cell, erythrocyte. So this is another very basic information, that what is the proportion of these blood cells in a healthy individual, what is the lifespan, the exact function. So RBC 120 days, the lifespan. So there is recycling of these blood cells, recycling of the blood cells continuously in our life. And that recycling of the RBC 120 days, the average concentration in a healthy individual 5 million per microliter of blood. Microliter means 10 to the power minus 6 liter, in about 1 drop of blood, the volume. Its exact role is transportation of oxygen. White blood cell. Now white blood cell, which we try to discuss to explain on immunity, antibody produced by white blood cell. So antibodies are produced by which cell? White blood cell. Within white blood cell, again these are different types that we are yet to discuss. One type we introduced the name lymphocytes that produce the antibodies. Now on an average, white blood cell in a healthy individual is about 7,000 per microliter of blood. We have written the lifespan as 1 week, but it will vary depending upon the type of white blood cell. Their lifespan can vary. Memory cells can even go up to years. They even go up to what? Years. So there is continuous. So once there is compartmentalization, selection, continuous production of the new cells, the new memory cells also. Because the vaccine should provide us what? Generally lifelong immunity. So this means memory cells could be selected and cloned for many years in your body. So their major role is immunity. Platelets. So platelets today we discussed. that their major role is what? It releases chemical signal to initiate the process of blood clotting. So this signal will start recruiting the blood clotting proteins, prothrombin, fibrinogen. Their average lifespan, that is about 10 days. Now, all of you know about this concentration is significant, particularly we correlate with the dengue virus disease. So platelet count, 1.5 lakh to what? 4.5 lakh per microliter of blood platelets. So this is about the composition and the lifespan proportion of these blood cells. Now next one is from where these blood cells are produced in your body. Hematopoiesis. The next discussion will be on hematopoiesis, which means synthesis of blood cells from the bone marrow, So we have blood stem cells in our bone marrow. We'll continue this topic again in the next class from bone marrow.